In the previous lecture, we discussed about the contribution of Louis Pasteur in the development of microbiology. Now today we're going to discuss about the contribution of Robert Koch in the field of microbiology. Heinrich Hermann Robert Koch, born on 1843, and he is a German general practitioner and a famous microbiologist. The Robert Koch play an important role to explain the various things which are very much important in the field of microbiology. So this Robert Koch is a founder of a specific causative agent of tuberculosis, cholera, and anthrax. And in the Pasteur contribution, he explained the each and every diseases caused by microorganism. But at that time, there is no any technique developed for the isolation of that microorganism which is responsible for the particular disease. But Robert Koch actually developed the various techniques which are very important and essential in the field of microbiology to isolate the pure culture as well as the causative agent of the particular disease. He is credited to be one of the founder of specific field of modern bacteriology, and that's why he is well known about the father of bacteriology, modern bacteriology. For this, he is also regarded as a pioneer of public health, aiding legislation and changing prevailing attitude about the hygiene to prevent the spread of various infections and various diseases. The Dr. Robert Koch was a pivotal figure in the golden age of microbiology, and it was the German bacteriologist who discovered the bacteria that cause anthrax, tuberculosis, and cholera. And his method enabled others to identify many more important pathogens. Thanks to his contribution to the field of microbiology, and he is sometimes known as a father of bacteriology, and a title shared with the Louis Pasteur. Koch's first important discovery was the anthrax, a disease that killing large number of livestock as well as some humans. And rod-shaped structure bacteria has been observed in the blood of infected animals, but the cause of the disease was still uncertain. Koch found that the disease could be spread by the blood of infected animal and hypothesized that it was caused by living bacteria. He developed sophisticated technique for the observing bacterial growth on microscope, on microscopic slides, and saw that the anthrax could form spores that survived dissection but produce more bacteria when put back into the moist environment. This explains how the contaminated soil could remain toxic for years. Although others had earlier determined that the germ caused disease, notably the Pasteur and Joseph Lister, Koch was the first to link a specific bacterium, in this case Bacillus anthracis, to a specific disease, anthrax. Koch learned that dates help to make a bacteria visible and identifiable under the microscope. Actually, the Koch developed certain techniques to observe the bacterial culture under the microscope, and on the basis of morphological characteristics, we can identify the shape and that bacteria, and publish the first photograph of bacteria by the Koch. Coach assistant Julius Petri and designed a shallow dish for culturing bacteria. That is a Petri plate. Nowadays we use Petri plate for the culturing of bacteria in a laboratory. And other of his assistant discovered that agar from seaweed made an effective medium. So whenever we prepare the nutrient medium in the laboratory to culture the bacteria, at that time we need the gelling agent to solidify that medium, and the coach assistants actually discovered that gelling agent nothing but the agar powder, that agar powder extracted from the seaweeds. Apart from this, a coach actually gives some postulate, and the tuberculosis was then responsible for one in the 7 deaths in Europe. Koch discovered rod-shaped bacteria called bacilli in the patient tissue, but needed more evidences that they were the cause of disease. He formulated four conditions that come into be known as a Koch postulate. First postulate of the Koch is that the organism was present in every case of the disease. Means any person infected by the particular pathogenic organism, so in that patient body, that particular pathogenic organism actually present. The second postulate is that the it could be cultured outside the body. Means from the infected person we can isolate that microorganism in the laboratory, and that same causative agent or that same pathogenic bacteria we can culture in a laboratory outside the patient body. Third postulate is that the inoculation of that culture caused the disease in an animal. So that same bacteria, whenever we inoculate it into the new healthy animal, so that bacteria is responsible to cause the same disease into the new animal or human. And the fourth postulate is that the organism could be found in that animal. So whenever we try to isolate the bacteria from that newly inoculated organism, that bacteria is same or identical with that of the first one. So the same causative agent must be re-isolated from the inoculated disease organism. We can re-isolated the bacteria which is responsible to cause the particular disease. The culturing the bacilli was difficult, but Koch eventually succeeded in growing them on coagulated blood serum and found that inoculating animal with the bacilli caused tuberculosis. He also found that the sputum from patient was the main way of disease, and through that sputum the disease can spread from one person to the another person, and this leads to the strategy to prevent disease by sterilizing clothes and bed sheets and prohibiting the public spitting. He went on to discover the bacteria that cause the cholera and demonstrated the importance of clean water supply to the prevent disease. Robert Koch was awarded in the 1905 with a Nobel Prize in Physiology or Medicine for his work on tuberculosis. Dear students, he investigated the anthrax disease cycle in 1876 and studied the bacteria that caused tuberculosis in 1882 and the cholera in 1883. He discovered bacteria such as anthrax, bacilli, tuberculosis causing bacteria, and as well as Mycobacterium tuberculosis, as well as the cholera bacilli. and Koch observed the phenomena of acquired immunity also. So he introduced solid media for culture of bacteria in a laboratory, and Koch pioneered the use of agar as a base for culture medium. He developed the pour plate method and was the first to use solid culture media for culture the bacteria. Apart from this, Koch also developed media suitable for growing bacteria isolated from the body, and because of their similarity to body fluids, meat extract and protein digest were used as a nutrient sources. The result was the development of nutrient broth and nutrient agar media that are still in the wide use of today. He also introduced methods for the isolation of bacteria in pure culture. For example, the spread plate method, then pour plate method. So, by using the different types of techniques, we can isolate the bacteria, specific bacteria from mixed culture, from a sample in a pure form. Because this technique is most important whenever you try to study about the particular bacterial species which is responsible to cause the particular disease. At that time, you should isolate that bacteria in a pure form. And due to the discovery of, or due to the formulation of the methodology by Koch, Robert Koch, for the isolation of pure culture, and from this method nowadays we can easily isolate the pure culture in a laboratory, and we can study about the that particular bacteria individually. It may be the morphological characteristics, it may be the biochemical, or it may be the molecular characteristics of that particular causative agent. And we try to understand the mechanism of that microorganism. He described hanging drop method for testing the motility. In the first semester, you know very well, in our syllabus there is one practical, and that practical based nothing but the demonstration of bacterial motility. As you know that in our nature, the some bacteria having the flagella, and due to the presence of that flagella, they can move from one place to the another place, and this phenomena known as a bacterial motility. But not every bacteria are motile. Some bacteria having ability to move from one place to the another place, but some bacteria do not have flagella. Due to the absence of flagella, they cannot move from one place to the another place, and that bacteria known as a non-motile. So such type of motility phenomena we can observe under the microscope due to the this technique formulated by this coach. The Robert Koch introduced staining technique by using aniline dye, because you know very well the staining is very most important method in the microbiology laboratory, because in microbiology laboratory we try to observe the various types of bacteria under the microscope, but we need to stain that bacterial culture. So the first time staining methods formulated by this Robert Koch, and with the help of aniline dye, he tried to stain the bacterial culture, and that bacteria we can observe under the microscope to study about the morphology, to study about the presence of flagella or absence of flagella. He invented the hot air oven and steam sterilizers, nothing but the autoclave, and also introduced method to find out the efficacy of antiseptic. So Robert Koch actually contributed in the development of microbiology by introducing the various types of methods and procedures which are very important to explore the microbiological world. Dear students, the third most important scientist in the field of microbiology, that is Edward Jenner. Edward Jenner was born on 1749, and he is a father of immunology. After the discovery of or after the finding of causative agent from the particular diseased person, the Edward Jenner worked for the development of vaccine uh, for various viral and bacterial diseases. So that's why this Edward Jenner familiar with that of father of immunology. Dear students, what is actually the immunology? Immunology nothing but the process or mechanism in a human body which can fight against the foreign material or foreign organism. Foreign material means it may be the dead or it may be the live. So, so the human being always exposed in the environment, and through that environment, uh, the various types of foreign material can enter inside the human body, and at that time some material or some foreign material recognized by the human body, that is known as immunology or immunosystem, and that immunosystem recognize that foreign material and try to diminish the action of that material by developing the antibody inside the blood as well as by the cell mediated immunosystem. And due to this type of mechanism, nowadays it is possible to develop the vaccine against different types of disease. So Edward Jenner was an English physician and scientist who pioneered the concept of vaccine, including creating the smallpox vaccine, the world's first vaccine discovered by this scientist. And the term was vaccine, and vaccination are derived from the variola vaccine. The term devised by Jenner to denote the cowpox. From the age of 14 years, he trained as a apprenticeship to Daniel Laidlaw, a surgeon, and in 1770 worked under Surgeon John Hunter at St. George Hospital, and returned to Berkeley in 1773 and practiced as a GP and surgeon. Edward Jenner died of stroke at the age of 73 on 26 January 1823.